So I have a few announcements before we get started. I'm recording this, yes. Okay, so there is a little bit of a change in this kind of the coming schedule. I do have a surgery on April 8th, so there will be a couple of weeks recovery for me. So lecture on April 14th will be pre-recorded. So I will pre-record the lecture during the spring break, and you can listen to it whenever you want, as long as you listen to it before April 21st. So just make sure that you do that. I will send an email reminder, but just wanted to make sure that you know that April 14th, there won't be a live Zoom. There will be a recorded lecture, and that I will post on Canvas. The other thing I want, due to this kind of change, on April 21st, we're supposed to have our second exam. What I will have to do is the exam will be on a Saturday that week, on the 24th. And Brigida and Redica, you will present on the 21st of April instead of the 14th, right? Because currently you were supposed to present on the 14th. Is that correct, Brigida?
Yeah.
Okay, so that will move to the 21st. And because of that, because we have a student presentation on the 21st, we won't have the exam on the same day. So the exam will be administered April 24th. Here's my question to you, and I can either open it the entire day on Saturday, make it available for you from 8 a.m. to midnight, and it would be, again, 85 minutes. Last time you needed this extra time, I can just make it 85 minutes. For the same amount of questions, it won't be more questions. And without having a live Zoom with me, so you won't be able to ask me questions. Or we can set a time on Saturday where we administer the same 85 minutes with you all and with me in case you have questions. So what is your preference? So I'm going to ask you, do you want it open all day? You can take it anytime, but the time will be 85 minutes no matter what. You just have to submit within this time, and I won't be there to answer any questions. So what is your preference? You can put in the chat your preference, and we'll go with majority. Please, all of you answer. Wow, we almost have unanimous. Okay, open all day. All right, so that's what I will do. I will just make sure it's open all day for you, but it is timed and you cannot close and go back and come back to it. So it will be timed 85 minutes. All right, so we'll do that. And on April 28th will be our last lecture. We'll finish up whatever I can finish up material, and we'll have the two last presentations, and that would be Chrissy and Leslie and Laura and Paul. Yep, good. All right. So with that, we have two presenters. Who will be starting presentation first, Holly or Devin?
Me, I'll be starting.
Okay, so presenter one is Holly and presenter two is Devin. Please make sure to fill the sheet. I sent it again to you in an email, just that you have it. And please make sure that you pay close attention. Hopefully you've looked through the papers. I have them on Canvas. And do ask questions at the end. It's really, I would like you to be engaged, um, and make sure that you have thoughtful questions for the presenters. With that, I will have Holly start sharing, or Devin start sharing, and then Holly, you go.
Okay, everyone can see and hear me?
Yes.
Okay, perfect. So hello and welcome to Proteins. Devin and I are going to be presenting our topic, which is food enzymes, specifically lipase and immobilization. So we're going to start off here with a few background slides to refresh your memory on concepts we've learned so far in lecture. Enzymes may be split into two classes, hydrolases and oxidoreductases, according to the type of mechanism they have. And enzymes have a wide range of applications in food, whether those be desirable or undesirable. For example, a desirable effect could be clarifying juice, which is a function of pectinase. On the other hand, something undesirable would be browning that you would observe in fruit, which is something that polyphenol oxidase is responsible for. So our presentation today specifically will fall into the hydrolase category and, as I said, lipase. All right, so in addition to that, we will be talking a lot about enzyme kinetics. So this slide will function as another memory refresher. And this figure shows how an enzyme fits together with a substrate. And this graph shows general enzyme kinetics. And we can see that as substrate concentration increases, reaction velocity also increases up to a certain point. And then Km is the substrate concentration at half of maximum velocity, which, if you remember, is an important characteristic of an enzyme as it indicates the affinity of the enzyme to the substrate. And then kinetics are dependent on a few key parameters listed here. So, the concentration of the enzyme substrate and any present inhibitors or cofactors can either slow or speed a reaction. And then additionally, pH, temperature, and ionic strength can affect the binding strength of the substrate in the enzyme. With that in mind, it's not surprising that enzymes present some challenges when utilized in industry. As I mentioned, environmental factors like pH, temperature, and ionic strength can affect enzymes and their kinetics. To name a few, large-scale processing can introduce high temperatures, foaming, and drying that denature the enzyme and cause it to lose its catalytic activity. This will in turn affect the efficiency of the production process, potentially causing extended processing time and reducing the yield of the desired product. And then additionally, enzyme denaturation will impact its recovery and its reuse. So all of these challenges contribute to the economic impact of enzyme utilization in industry. One modification that has been proposed to address these challenges is enzyme immobilization. This process attaches an enzyme to or within a matrix to theoretically enhance its interaction with the substrate and increase its activity, stability, and reusability. Three main immobilization techniques have been reported in literature, including carrier-bound attachment, encapsulation, and cross-linking of enzyme aggregates. And despite reported success with this technique, most research has been done for non-food applications. So therefore, there is a need for development of food grade or edible immobilizations for potential application in the food industry.
So our specific papers that we chose, we wanted to break it up. We are looking at increasing the actual activity of the enzyme. And then the other paper is going to look at increasing its processing stability and its ability to be reutilized. So our first paper is taking immobilized lipase, comparing it to free lipases for cheese production. And our other paper is going to be looking at its ability to make omega-3 fatty acids over many cycles. So the first paper here is an enhanced fatty acid flavor release in low-fat cheeses by mobilized lipases. They were looking to do this because the paper saw that there needed to be more immobilization techniques for lipases for food applications, and also that low-fat products tend to have or need flavor enhancements. And one way to do this is by releasing free fatty acids using lipases. So the objectives were to create an immobilization technique for lipases that would be able to stabilize a milk fat emulsion and also increase the efficiency and release of free fatty acids and potentially a final cheese product. So their design here is the immobilization technique was their first step, and they took alpha-lactalbumin, partially hydrolyzed it with enzyme to create a nanotube. And this nanotube is what they're actually going to cross-link the lipase to. The nanotube they chose was because it could help stabilize emulsion and get the lipase potentially to the emulsion interface to interact with the oils. And they can verify that the lipase is going to cross-link using fluorescence scanning. Once they created their immobilization technique, they checked the activity of the lipase by comparing it to a solution with free lipase versus immobilized lipase and adding para-nitrophenol palmitate to it to see how much hydrolysis occurred so they could check the activity. And then they also put the emulsion into a cheese formula to see how much overall free fatty acids formed within the cheese. So here on the left, we can see this is the nanotubes they created after the partial hydrolysis. And on the right is after the lipase was actually cross-linked to it. So they're just showing that the lipase actually clustered to these nanotubes. And to check how much of the lipase was actually cross-linking with the nanotubes, they attached the CY5 compound that absorbs at 660 nanometers. And then they centrifuged all of the not cross-linked lipase out. And what they saw was that about 16.67% of the lipase was actually cross-linking, which seemed relatively low, but they didn't really talk about possibly increasing that number at all or what their thoughts were on the efficiency of the immobilization technique. And on the right, they checked the zeta potential of the nanotubes before and after to show that the cross-linking was actually occurring, and so the zeta potential became less negative. So lipase activity, this is where they're taking the immobilized lipase and comparing it to a solution of free lipase with a para-nitrophenol palmitate. So they're looking at the concentration of enzymes and then the amount of product being formed to create that Lineweaver-Burke equation. And what they found is that the immobilized lipase in general was able to produce a lot more of the product, which means that the lower Km and higher Vmax means that the lipase has a higher affinity for the substrate and will produce more of the compound. So now we're going to the emulsion aspect of it. On the top there, we can just see that's just the oil and water emulsion they created. And over time, as hydrolysis occurred, the oil broke up and coalesced. On the C line there, that's where they actually labeled the lipase with green to show that the nanotubes were able to bring the lipase to the outside of those droplets to interact with the most amount of oil at that interface. They created two different droplet sizes for their emulsions, doing two different techniques, and one of them being a smaller droplet size, one of them a larger size, just to show that if you increase the surface area of the interface by making the oil droplets smaller, then the lipase will be able to interact with more oil and produce more free fatty acids. So see here, we can, what they compared was just a lipase emulsion without immobilization, and then two immobilization emulsions, one with the large droplets and one with the small droplets. So without immobilization, the free fatty acid release was considerably worse but the two immobilization solutions had similar ones, but creating a smaller droplet size emulsion ended up producing the most amount of free fatty acids. Now back to cheese. So up on here, I think we've seen images of this before, where the green is going to be the protein mesh, the red is the fat, and the blue is going to be the labeled lipase that they added to it. So they're comparing a full fat cheese to a non-fat cheese to a low fat cheese with the immobilized lipase and just a normal low fat cheese. And what they wanted to show was that the low fat cheese with and without the immobilized lipase had the same fat content that we can see in I there. But in J, we can see that had a considerable amount of more free fatty acids that were released due to the immobilized enzyme being added to the formula. So in conclusion, they found that immobilizing the lipase with this technique did bring lipase to the emulsion interface and increase the activity of it considerably. I would have liked to see them show more of their GCMS work for their overall free fatty acid content. They didn't mention the actual protocol or show any of the chromatographs. And I think that also more of the articles should have been focused on actually improving the food itself. They showed that lipase activity outside of the cheese was increasing and behaved better, but I would have liked to see that it actually changed the cheese considerably to a point where people would consider using this product. And maybe even add some sensory evaluation to show that the extra processing would be worth it for them.
Okay, so our next manuscript was published in Food Chemistry in 2019. This is the title in the left box, and it's long, so I'm not going to read it. But in essence, this study looked at producing omega-3 esters from chia oil through different types of immobilized lipases. And omega-3 is desirable to isolate from foods as it has positive impacts on health. And traditionally, fish are one of the great animal sources of omega-3 fatty acids, but overfishing has caused the need to seek out a more sustainable source. And chia seeds are a plant source of omega-3 and contain the highest known percentage of ALA omega-3, which made it a good starting material for this study. And as emphasized throughout this presentation, enzyme stabilization is needed for industrial applications to reduce processing costs. So that's another reason that they wanted to do this study. So these are the objectives. Again, they wanted to synthesize omega-3 esters from chia oil using four different types of immobilized lipases produced in their lab, and then compare their results to 2 commercially available immobilized lipases to see how the different types of immobilization affected the efficiency. Specific things monitored included the rate of omega-3 production by the enzyme, the purity of the product, and also the reutilization of the enzyme. So here is the experimental design and the methods. They started with lipase immobilization itself, shown in this figure. Tested modes of attachment included hydrophobic adsorption, chemical amination, and something called pegylation of each of those types. So in total, they had 4 immobilized lipases. And pegylation refers to coating of the enzyme carrier with something called polyethylene glycol to make an additional barrier. And then after immobilization, they performed the reactions with the modified lipases. There were 2 methods of analyses, Hplc and GCMS, and I'll touch on the specifics of each of those in the upcoming slides. And then lastly, they looked at the recovery and reutilization of the immobilized lipases to assess the impact of the immobilization technique on enzyme longevity. As mentioned, Hplc was used to identify the presence of the fatty acid ethyl esters relative to the tri, di, and mono acylglycerols to see if the enzymes were successful in hydrolyzing the starting chia oil. So this is an example chromatogram given in the manuscript. At the top of the figure, labeled A, is a standard mixture of lipids. The starting chia oil, labeled as B below that, has a peak corresponding to triacylglycerols. And then as the reaction proceeds, lipase hydrolyzes the triacylglycerols, and you start to see a distribution of peaks corresponding to the fatty acid ethyl esters in the products. And then next, gas chromatography in tandem with mass spectroscopy was used to identify specific fatty acids within the product to ensure that the omega-3 fatty acids were indeed present. And they didn't provide any example chromatograms, but simply claimed that the product for all the tested lipases contained equal amounts of omega-3 relative to their starting material. So then they claimed that the reactions were successful and each enzyme functioned to produce the product of interest despite or regardless of immobilization technique. And this is where we start to see some differences depending on how the enzyme was immobilized. So on the x-axis here, we see the type of immobilized enzyme tested, with the two commercial ones on the left side and then the lab produced ones on the right. So the y-axis is the fatty acid production at 4, 8, and 24 hours of reaction time. The adsorbed lipase was closest to the commercial samples, so that's that one right here. In terms of kinetics, producing close to the maximum amount of fatty acids after 4 hours of activity, and even produced what they claim to be 100% after the full 24 hours. And then the other immobilization techniques had varying effects. The pegylation I mentioned did not interfere with the reaction in the sense that they were still able to identify omega-3 in the product, shown right here in this bar. But it definitely slowed the kinetics because you can see it took a lot longer to start seeing those increased amounts in the product. Lastly, they looked at reutilization of the enzymes. So these figures show the change in percent enzyme activity on the y-axis as the enzymes were subjected to increasing reaction cycles on the x-axis. The figure at the top of the slide compared the adsorbed lipase to the commercial samples. The commercial samples, as you can see, had very poor reusability, while the adsorbed lipase performed better. And then at the bottom of the slide, this figure looks at the stability of one of the immobilized lipases, the aminated and pegylated one. And this time they tested it over 10 reaction cycles. The authors claimed that this one had the best stability because 82.1% of the original activity was retained even after 6 reaction cycles. However, as shown on the previous slide, this specific one had very poor kinetics. So it wasn't necessarily helpful to show that it has good stability if it's going to be very slow. And then to summarize this manuscript, the authors concluded that the omega-3 production by immobilized lipases is feasible at an industrial scale. The authors recommended the pegylated lipases as these ones showed the best stability. However, there was no control lipase in this experiment, so it's kind of hard to gauge the actual improved kinetics or stability of the immobilization when all the results are relative to each other versus like a control. And then as shown on the previous slide, they only used a pegylated lipase for their extensive stability study, even though the adsorbed lipase was the one with the best kinetics. Other cautions include the sensitivity of detection, as they claimed in one of the reactions that it's 100% efficient, which sounds a little fishy to me. And they also failed to provide more evidence of their claims and seemed to show the same data in multiple different figures throughout the manuscript. So therefore, my recommendations are to address those things by using a control lipase and also reduce that redundancy and provide more evidence to back up their claims.
We thought overall that these papers did find that immobilization techniques can increase the activity of an enzyme, and they also do have the ability to increase the stability and reutilization of them. And both of them sought to find immobilization techniques that would actually be food safe, whether it's being consumed in the food or reutilized in the food. And that seemed to be a driving force for both of these papers. Improvements we thought were compare the final product to like what's available on the market right now, especially with like the cheese, actually proved that doing these extra processing steps and adding enzymes with potentially having to immobilize it yourself. You want to prove that your product's going to be that much better and more enhanced. Also, if these immobilization techniques can be scaled up, how feasible can they be for industrial use if they're going to have to do it themselves? Because a lot of immobilization techniques are all over the place right now. What specific compounds they're doing, a little bit of variance into how they're actually immobilizing it. So overall, the study should focus a little bit more on industrial feasibility.
Cool. Thank you, guys. That's a good presentation, good summary. All right, we'll open it up to questions for the cat and dog, the pictures.
Reggie.
Okay, don't be shy. You can go ahead.
I'm looking at the synthesis of omega-3 paper. It's page 436, just above the section 3.2. It says, pegylation process did not produce a loss in the activity. I guess, what loss were they referring to? And what that just seemed to just, were we supposed to expect a loss to happen? It wasn't mentioned before. Maybe I'm missing it. The sentence just threw me off.
Can you say your question again, Paul? Sorry.
It's on the omega-3 paper.
Yeah.
Page 436, right above section 3.2. The pegylation did not produce a loss, and I guess I wasn't really expecting them to discuss a loss if there was any. They didn't make it like hypothesis or anything about a loss. So I just
Yeah,
what do they think that would happen? Like,
I think they were a little bit, I was also trying to look into this more, like what they thought their reasons were for the different like catalytic activity that they saw. Earlier on in that paragraph that you're referring to, they talked about some like sterics and polarity of what they thought the pegylation introduced. So I think that, like it says, the pegylation process gives a high polarity of the lipase surface. Therefore, a smaller amount of lipase per gram of support is being used. So I think that because pegylation introduces more mass in your enzyme, they're using a smaller amount of lipase of the immobilized enzyme per gram of support. So that's going to slow down your kinetics because there's going to be less catalytic activity there via the pegylation process. So that's why they're claiming there's not a loss of activity in the sense that the lipase is still functional. It's just going to be slowed down because it's kind of encapsulated by a barrier. So it's going to take a longer time for the substrate and the enzyme to interact and contribute to the kinetics of the reaction. That was my understanding, if that makes sense.
I think it's a good understanding, Holly, of that. And I just have a follow-up before I open it also to others. So they saw less kinetics or slower rate of the reaction. Is that that's due to having less enzyme per se, not necessarily the same amount of enzyme, but it's just the presence of pegylation just prevented the substrate from interacting. So less amount of enzyme total compared to the others.
Yes.
Okay, so would that, okay, if we consider less amount of enzyme and you were able to still get the same productivity after 24 hours, because after 24 hours, you got to the same amount of production. would using less enzyme, you're putting less money in to put less enzyme, would that be sufficient to say it's okay to have longer processing time, and then a more stable enzyme that can be reused? So could that be the reasoning for selecting that one pegylated enzyme, to look at stability and say, well, since you're using less enzyme, then potentially overall that's more efficient cost-wise?
Yeah, I think in theory that makes sense.
But
Again,
maybe they didn't make it clear.
yeah, in theory that makes sense. They didn't necessarily touch on that. And again, all their results were done like on a bench scale. So it's hard to like think about this in industrial picture if their results are kind of limited to a smaller scale. But yeah, I think that's a big concern in industry, is like, will we see these same effects, observed effects, when we scale up? If so, then yeah, it might make sense for them to save that cost in terms of enzyme amount. But yeah, it's hard to say, I guess.
Yeah, they need to test it obviously on scale up.
Yeah.
And the commercial sources, were they immobilized or were they free? Okay,
They
so
were immobilized, but they
were
didn't,
also immobilized. Okay.
yeah, they just didn't say like by what method of immobilization.
Mhm. Okay.
For the
Yeah.
article, they're interested in the recyclability. So a lot of their conclusions at the end of the paper were more concerned about it being able to be reused over multiple cycles, as opposed to, yeah, just how productive it was.
Okay.
I was also thinking about like enzymatic activity versus like stability. And I was just wondering, when it comes to reutilization, like do any of you know how do they actually reuse the enzyme? Do they just immobilize it again?
So the actual immobilization technique allows them to draw it out. So a lot of the other techniques they use in like biofuels are actually have magnetic particles in the immobilization carrier, so they can easily centrifuge or bring it out of the oil or whatever matrix they're doing. So there's a lot of different ways you can bring it out, but the immobilization part of it is that it's linking all the enzymes to something you can pull out.
Yeah, pull out, reuse, or the product goes through and then you put in another product. So that's kind of the idea, so that you don't put the enzyme directly in a batch process and then you lose the enzyme potentially. That you can't reuse the enzyme.
But for the cheese paper, though, that the immobilized lipase ends up in the cheese.
Okay, other questions?
I have a question. So, and this, Devin, this might have fallen kind of into the critique that you already mentioned, but it's just something that's been floating around in my head. Of, you talked about with the first paper, like, and even with the second one too, figuring out the like financial feasibility of doing this, considering things like processing time and all that. But I'm wondering how much like the materials or things for like the nanotubes or some of those other, like what they're actually immobilizing it with, like, does that come out to be cheaper than an enzyme? And if this might be kind of out of like left field, I don't expect you guys to know, but it's just like floating off in my head of like, are these materials common and less expensive enough that like replacing or using them to immobilize enzymes and getting to use less enzymes, like if there would actually be a financial benefit?
Yeah, especially when there's multiple steps and processing steps for immobilization. It seems like that could be just more and more of a consideration. I don't know exactly if they actually compared, how much more money you make on cheese with immobilized enzymes. I don't think they didn't really get anywhere close to that. But I imagine on something where you're actually producing a product like the omega-3 article, where you can reutilize them, and maybe it would be easier economically to figure out just like how much more money you're going to make with that extra step. I guess that's why I talked about, especially in the cheese article, if you're looking to enhance the flavor of it, what is the enhancement of like the sensory qualities that's going to make the cheese better? Because that's a little bit harder to relate back to the economics of it. Like how much more is that cheese worth now?
Right.
Yeah, and I thought that was a good thing that you guys pointed out of like, I know you both did it together, but like, is this cheese actually better?
Yeah.
Or even the same? Does it matter?
Yeah, it doesn't matter. Yeah.
More questions?
Yeah, with the cheese paper, for these nanotubes, it looks like the nanotubes are just alpha-lactobulin, and then the lipase was immobilized on that, so it's a protein immobilized on protein.
There's a glutaraldehyde that covalently bonds them to each other. That's that cross-linking that's actually going on.
Okay.
Yeah, that's just maybe they just mention it in between steps, I think. Yeah, but so they take that lactoalbumin and then they're partially hydrolyzing it with an enzyme. That's the first step. That actually creates the tube.
So Devin, did they explain how actually they formed these nanotubes and how they confirmed the formation of these nanotubes?
I just remembered that they did hydrolysis, then they added the calcium ions to it, and then they just had that picture of the tubes and the.
How did they take that picture? Or was it an animation, not the real picture, not a real image?
Oh, I think that was the CLSM photo, if I remember right. That's what they were doing for a lot of their.
So is this drawing the blue and the red is CLSM? It seems like it's an animation.
Which one? The blue and red?
And the nanotubes that you're showing in blue, how did they confirm the formation of nanotubes? By imaging?
Oh, it's, as far as I can tell, they just had an image of the nanotubes and that was it. They just did some processing steps that they found in another paper to make nanotubes. And I think between those two things, that's all.
Okay, so they referenced another work to develop, to produce these nanotubes, right?
Yeah, yeah.
Okay. All right.
They called it like self-assembly nanotubes there, yeah.
Okay. One thing you said, Devin, about they tested the lipase activity outside of the food system, but they did also test it in cheese, didn't they? Because they looked at the cheese as well, and they looked at about how much fatty acids were produced in the cheese.
Yeah, all they did for the cheese was they just did total fatty acid composition at the end of the cheese production. And I would have liked to see more of just like what was the fatty acids, how is it changing? If they want to,
but they did use their immobilized enzyme in the cheese.
Yeah, but their kinetic work was outside of the cheese production.
Oh yeah, the kinetic outside, but they kind of confirmed the production of free fatty acids in a food system in the cheese.
Yeah.
Yeah. Okay. One last question from you guys. I really would love to hear more questions.
Yeah, I have a question. It was originally for Devin, but I guess it could go to either. Did they do any sort of like long-term studies on like sensory or shelf life or flavor profile or anything using these types of enzymes, nanotubes?
That was your criticism, right, Devin? There's no sensory done.
Oh,
so they weren't sure if like the effects actually improved their end product, which is the point of the whole study seemingly, is to make a better low-fat cheese, but they didn't actually test to see if they made a better low-fat cheese because they didn't conduct any sensory studies on it.
Yeah, that would be a good follow-up. Feasibility studies would be good. Cost feasibility in terms of processing feasibility as well as cost efficiency would be nice. But it's good, I would say they're good basic type of start information. that could potentially lead to scale up. I think it's not as much as flaws, more follow-up studies need to happen. That's how I see it. I think it's good, valuable work, basic work. Yeah. Okay, well, that's good. So what we'll do, we'll take 5 minutes break. I will come back at 3:40 and we'll do 60 minutes lecture. Okay, let's come back. All right. I see there were a couple of questions in the chat that I missed, but Devin answered one and is answering the other. Okay, good. Thank you. All right, so let me share my screen. All right, so I have later on recap kind of questions embedded in a table format, but I just want to go back to where I left off. We finished talking about casein proteins and we started talking about whey protein. So I will quickly review this slide and move on to remind you of where we were, and then we'll do the recap in a form of a table. Okay, so whey proteins represent 20% of the total proteins in milk proteins. And we said they have mainly the main functional proteins, and the larger components of whey proteins are alpha-lactalbumin and beta-lactoglobulin. So these are the main proteins that contribute to the functionality of a whey protein ingredient. So they do not associate in a quaternary structure like the casein associate in a micelle. They mostly are monomers or dimers, or they could be octamers based on just pH changes. And I'll talk more specifically about that in a minute here. And in terms of structural configuration, they are globular and compact structure and have very ordered secondary structure. So for alpha-lactalbumin, you have mostly alpha helices and one strand of beta sheet. And for beta-lactoglobulin, you have mostly beta sheet and a little bit of one alpha helix really, and a little bit of random coil in both of them. So the difference in the secondary structure, when you have more alpha helix than beta sheet, that means the denaturation temperature is impacted. Beta sheets are more heat stable. So you'd see that beta-lactoglobulin denatures a slightly higher temperature than alpha. And also one important characteristic is the cysteine residues. So in alpha-lactalbumin, you have cysteine, 8 cysteines and 4 disulfide linkages. So all of the cysteines are involved in disulfide linkages. So if you take alpha-lactalbumin and try to form a gel with it, the gel is not as strong or as elastic because you don't have disulfide interchange. You don't have this trigger that causes disulfide interchange. The trigger that causes disulfide interchange is to have a free sulfider group. And the beta-lactoglobulin has a free sulfider group. It has 5 cysteine residues. 4 of them form disulfide linkages. So you have 2 disulfides here. And then you have this free sulfider group that is embedded within the interior moiety of the protein. Upon denaturation and opening up, becomes exposed and reduces other disulfide linkages and causes disulfide interchange through the redox type of reaction. So it's a very important component, functional group in the gelation mechanism of whey protein. Oh, I showed you this slide also last time. And as a reminder, I show the solubility curve here. So being a globular protein with high surface hydrophilicity, so it's very hydrophilic on the surface with one pouch of hydrophobic residues, this makes it a very soluble protein. And even at around its isoelectric point before heating, it's still a soluble protein, even at zero net charge, because you don't have enough hydrophobicity on the surface to draw the proteins together. But once it's heated, then the protein denatures and opens up. So at zero net charge, you have more hydrophobicity on the surface that will cause protein-protein interactions, and then you lose solubility. So this is very characteristic of whey protein. It is different than other proteins we'll cover. Yeah, so basically I covered all of what's on this slide. And last time we stopped here. And here's more specifics about what heat does. So the stability or thermal stability of beta-lactoglobulin per se. So when you pasteurize the milk, for example, beta-lactoglobulin is the nature. When you formulate with whey protein, put it in a beverage, in an acidic beverage, a high protein beverage, also you pasteurize, you're denaturing the proteins. So over time, basically, even whey protein, although it's very soluble, but because these beverages or milk or what have you been pasteurized, over time, the denatured protein will, start getting in contact with each other and form aggregates and precipitates. So shelf life is impacted by that. Also, the denaturation, let's say if you heat the milk and the protein denatures, and then you're trying to form a cheese, then what happens is beta-lactoglobulin is going to interact with casein, might slow down the enzyme hydrolysis by chymosin, enzyme hydrolysis of the casein into a glycomicropeptide and the para-casein. It is also pH dependent, so heating at close to the isoelectric point causes denaturation sooner or at lower temperatures, because then you have less stability when you are around or close the isoelectric point. And when you have higher net charge, you have a maximum stability. So if you try to determine the denaturation temperature of whey protein, it's impacted by pH. So if you're measuring at different pHs, you might get different temperatures at which the protein denatures. So stability decreases with increased pH, because increased pH can also cause an increase in net negative charge, especially not only with the amine groups losing their protons, but also your hydroxyl groups on some amino acids and sulfide groups can also lose protons and becomes excessively negative charge, and that causes repulsion within the protein and further destabilization at lower temperatures. And we see this not just with whey protein, we see it with other proteins, and especially we notice it in our lab when we try to extract protein at higher pH, that causes denaturation. So at lower temperatures. So basically, denaturation here is impacted. So it's not just characteristic to whey, but these are the conditions characteristic to whey, but it applies to other proteins as well. Around the isoelectric point, you get protein denatures at lower temperature. At higher pH, the protein denatures due to repulsive negative charge. And this happens more at alkaline pH than acidic pH due to additional net negative charge to hydroxyl groups and sulfide groups. So just try to remember that. You don't have to remember the actual pH values. You just want to remember that how the denaturation is impacted by pH, around the isoelectric point, around neutral pH, and around alkaline pH. So in terms of stability to pH, so it's stable at acidic pH, and the molecular association, as I mentioned earlier, is impacted by the pH. So beta-lactoglobulin form monomers when the pH is under 3.5 or above 7.5 due to charge, excess charge, and therefore then they don't associate with each other. Around pH between 5.5 and 7.5, often we find beta-lactoglobulin as a dimer. So let's say we're trying to separate whey proteins by size exclusion chromatography without adding any reagents or detergents that would cause dissociation. And if it's a neutral pH, we see a peak around dimer, which is around 36, 37 kilodalton. Because whey protein beta-lactoglobulin is around 18 kilodalton. But when we run it on a gel, we always see it as a monomer because the STS page gel that we run, we add STS and it kind of breaks down non-covalent interactions. So there's no association. So we see it as a monomer around the 18 kilodalton. Okay, so it forms an octamer at pH around the isoelectric point. So if you try to put beta-lactoglobulin in solution and you lower the pH, let's say to 4, 4.5, it's still going to be soluble and dispersed, but it's just going to be a little bit more associated. So usually it forms an octamer around that pH. Above high alkaline pH, we already mentioned that you would have repulsion and it will open up, so it will be expanded monomer. That means unfolded protein. So alpha-lactalbumin, we said already, it's also compact. It has mostly alpha helices, does have some random coil, 1 beta sheet, and the 8 cysteine residues, all involved in disulfide linkages within the protein itself, so intramolecularly. It is a calcium metalloprotein. That means it has tendency to bind calcium. So in the presence of calcium, it has better stability because the calcium is a divalent element or metal element, and it causes interactions within the protein by divalent, by ionic interactions, so enhances the stability. So if calcium is depleted or removed, the thermal stability or the stability of alpha-lactalbumin will be reduced. In terms of biological function, it functions as a synthesis enzyme. Well, it's an enzyme regulator for the synthesis of lactose. Okay, that's a biological function. So, like I said, it's more heat sensitive than beta-lactoglobulin due to alpha helices, but denaturation can be reversible because if it's present alone and there's no beta-lactoglobulin, that means there's no free sulfide group that can cause disulfide interchange. So if we heat it and then let it cool, and then it might come back to renature because the heat would mostly break hydrogen bonding and electrostatic interactions, but then upon cooling, they might form again. But if you have beta-lactoglobulin, and remember that if you have beta-lactoglobulin and alpha-lactalbumin together, and then they're both denatured, the sulfide group in beta-lactoglobulin isn't going to cause disulfide interchange and interfere with alpha-lactalbumin disulfide groups and also reduce them and have them oxidize with other sulfide groups in another molecule, causing that interchange and polymerization. And like I said, when you remove calcium, what happens, especially at low pH, when you have positive charges, then the protein becomes more heat sensitive. This is a summary table, a nice FYI table comparison between whey and casein in terms of molecular weight, number of amino acid residues, lysine, proline, phosphorylation, bisulfide, secondary structure. So it's kind of a nice comparison between the two. And here's a table where we're going to have some review between differences between casein and whey, which could be on an exam. One day I might ask you in one exam, tell me how casein and whey are different in certain properties. So let's discuss together, and this way you also can remember a little bit about casein that we covered last time. So in terms of amino acid distribution, and what I mean here is the hydrophobic residues versus hydrophilic residues. How are they distributed or differ in this distribution between casein and whey? Okay, so go ahead, either in the chat or unmute yourself.
So casein has like that better hydrophobic to hydrophilic balance, right? That's why they make great emulsifiers versus whey is more globular.
Yes, but more specifically, they're very amphiphilic or amphipathic. That means you have concentrated hydrophobic residues on one end versus another. So whereas in whey, you don't have the amino acid polars and non-polars are kind of like evenly distributed. You don't have like a pouch of very hydrophilic and a pouch of very hydrophobic. So that is what is meant by that question. Molecular structure or configuration of the monomers.
Casein are unordered, but whey proteins are completely ordered proteins because of the high amounts of alpha helix and beta sheet structures they have.
Yes, absolutely. So we have the molecular structure of casein. You have little secondary structure and tertiary structure. It's really mostly random coil, and because of the proline in there, it makes it more open structure rather than ordered and globular structure as in whey. So you have very ordered structure, secondary structure, alpha helix, beta sheets, and then a very compact globular structure. All right, so in terms of heat stability, do you remember the stability of casein, the thermal stability of casein and formation of gel? So we can talk about thermal stability first.
High thermal stability, they have high thermal stability.
Yes, so the caseins have high thermal stability. They are stable up to 135, 140 degrees for a few minutes. What about the casein gelation? How do you think, under what conditions casein gel? Well, you should know this. Huh?
Is it under cold conditions?
Say that again?
It's a little bit under colder conditions, cold temperature, they form reversible gels.
What other than temperature is really important for caseins to form a gel? Think of yogurt.
pH. Do they need like the kappa casein or something to help gelation?
pH, yes. So, well, we're getting to the kappa casein, Nigel. So pH, so at the isoelectric point, they aggregate with each other, and that's how yogurt forms, and yogurt is a gel. Kappa casein is important, and that's enzymatic gelation. So casein gets hydrolyzed by the chymosin, loses its stability of the micelle, and the micelles come together via hydrophobic and calcium phosphate bridges, forming the curd, which is another form of gelation. You also have age gelation we talked about. So let's say you have the milk is stored in the cooler, in the refrigerator, but you have endogenous plasmin and microbial enzymes that could, non-pathogenic microbes might be there and produce proteases, and beta casein would hydrolyze and then gel over time, forms a gel over time. So an age gelation, so that's called age gelation. So you have pH gelation, enzymatic gelation, age gelation. Heat is not, thermally induced casein gel is really not something that can easily happen due to the heat stability of casein. Now, in terms of whey, then, so it's heat labile, and we talked about that. So it denatures at lower temperatures, so we can produce thermally induced gels. So basically denature the protein, expose hydrophobic groups, expose the free sulfide groups at a particular protein concentration, which we'll talk about in a minute here, causes that, protein-protein interactions, and then eventually formation of the gel, thermally induced gels. Okay, so in terms of association, how do the caseins associate compared to whey? What do caseins form?
They can form self-association.
Yes.
Because
so
of high hydrophobic residues they have, they are too prone to self-association.
They form micelles?
Micelles.
Yes.
Yeah, exactly.
Yes, so they form micelles. So basically, the caseins form micelles. So you have all of the 4 caseins associating with each other via hydrophobic interaction, via calcium phosphate bridges, and also they are stabilized by the kappa casein mostly on the surface due to the glycomacropeptide or the glycomacropeptide portion of the kappa casein. How about whey? We just said, how do they associate? Do they associate or they don't associate?
They associate, but under heat conditions and near to isoelectric point, like for example, BLG or beta-lactoglobulin associate at isoelectric point or around pH 4-5. They are in the form of dimer, octamer.
Yes. So yes, and I would love to hear from the students so that they can get participation points for answering questions. So yes, so micelle formation, and then whey proteins usually are dispersed. They don't form stable quaternary structures like caseins, but depending on the pH, they can be monomer, dimers, or octamers. Okay, so enzyme sensitivity. Which one of these would be more sensitive to enzyme hydrolysis based on what we talked about?
Caseins?
Caseins, yes. So caseins are susceptible to chymosin hydrolysis, are susceptible to plasmin hydrolysis, which is an endogenous enzyme in milk, and also bacterial proteases that might grow under refrigerated conditions that are non-pathogens. So even if you pasteurize the milk, you still have non-pathogens in there that can grow during refrigerated storage and produce proteases that would hydrolyze. And mostly they attack the beta casein. Whey protein resists hydrolysis. Why? As in its native form, though, please remember, in its native form, it's globular. So globular proteins resist hydrolysis because of the access of the enzyme to binding sites. So unless it's denatured, then it becomes more partially denatured, not highly polymerized, partially denatured, unfolded, not polymerized. It will be more susceptible to enzyme hydrolysis. Okay, so in terms of surface adsorption and emulsification, how do they differ?
Casein is a better emulsifier due to their distinct regions of hydrophobic and hydrophilic. It's smaller, it has open structure, so it's more flexible.
What is more flexible?
Oh, casein.
Okay. Yes. So casein is more open structure, more flexible, amphiphilic. So it has high surface adsorption, whereas whey protein has low surface adsorption, has low, the balance, it doesn't have the same balance of hydrophobic to hydrophilic. It's mostly hydrophilic in the cell. But if you denature it, if you cause opening up of the structure, then you have better absorption. Okay, so in terms of solubility, how do they differ?
Pam, I had a question before we go to solubility.
Yes.
Whey also forms a thicker film around the air droplets as compared to casein because they're smaller and they go to the surface faster and more of
Yes.
them. Okay.
So in terms of stability, if they get to the surface and if they form an emulsion, it would be thicker and stable. So it would be good for an emulsion to have a mix of proteins. You want to have those that have high absorption and also those that you can have more protein on the surface. So then you can get the best of the two worlds. Okay, so going to solubility. How do they differ?
Whey is more soluble.
Yes. So casein is very sensitive to pH, regardless of heat or denaturation. At 4.6, the glycomacropeptide that is stabilizing the micelles loses its charge, the micelle loses its stability and gels right away. So no heat really is needed to form that kind of, to cause that precipitation. In case of whey, it is very soluble at low pH and it remains in solution and gives you clear solution, which is desirable. And also around the isoelectric point, unless of course you heat it, denatured around the isoelectric point, you lose solubility. Conjugation. So is the proteins, meaning here, are they all made up of amino acid homoproteins or heteroproteins? How do the caseins and whey differ in that case?
So we have the glycoprotein with casein.
Yes. Yes. So we have glycoproteins and we also have
phosphorylated.
Yes, so we have glycosylated proteins and phosphorylated proteins. In whey, we have none. Okay. Sulfur residues. How do they differ? In terms of sulfidyl groups and disulfide linkages.
So in casein, there would be more free sulfhydryl groups compared to whey?
Actually, in casein, you have low sulfhydryl groups in general, and you have low sulfur containing residues. Mainly you have methionine. You only have one disulfide linkage in kappa casein and one disulfide linkage in alpha S2. Whereas whey, both alpha and beta are rich in sulfur groups. And you have more disulfide linkages. You have 4 in alpha lactalbumin and 2 in beta lactoglobulin and 1 free sulfhydryl groups. So this is a good study table. It's good to differentiate main differences between casein and whey. So now, of course, this is recorded. If you didn't have time to write everything, you can go back to the recording and find that slide. So I'll talk a little bit about the functional properties of casein and whey. But before we go there, just a reminder, functional properties are related to molecular characteristics. So can you name a few molecular characteristics that will contribute to functional properties? Just kind of like a flashback to functionality and structural molecular characteristics.
Amino acid composition and sequence?
Yes, amino acid composition and sequence impact structural configuration. So structural configuration is important. What else is important in terms of functionality? We've said it a lot of times.
Surface hydrophobicity.
Yes, so surface activity. So whether it is charge or whether it is hydrophobicity. So surface activity is important, net charge is important, configuration of the protein is important in terms of the secondary structure, the tertiary structure, and the quaternary structure. So all of this, these are the main contributors. Of course, there is also the molecular weight if we want to think about more characteristics, but these are main molecular characteristics that impact functionality. Okay, so if we look at the emulsification properties, and if we consider casein, we've said that multiple times, high surface activity due to the being amphiphilic, absorb readily. So you want to remember this figure we showed when we talked about emulsification functionality. So you want the protein to absorb easily. That means in order to absorb at the interface, which is the interface between an aqueous and the oil phase. So you need to have that balance between lipophilic, hydrophilic properties. So you need absorption at the interface. You need the protein to be flexible so that it unfolds, opens, changes orientation to orient the hydrophobic components to the oil and the hydrophilic to the aqueous. And then you also want the ability to form a film. So you want protein-protein interactions as well to form a film around your oil or water, depending on the type of emulsion. So for that, casein has high surface activity, can absorb at the interface easily, is flexible enough to unfold, and can have enough hydrophobicity to be able to form this, the film, the thin film or whatever that film that holds the water droplet or the oil droplet. In terms of emulsification properties of beta-electroglobulin specifically, it has low surface activity, we already said that, but it's impacted by pH, it's impacted by disulfide linkages, it's impacted also by temperature. So at low pH, it's not going to emulsify well for you. It's compact, it's rigid, it's very hydrophilic on the surface. However, if we reduce the pH if we bring the pH to around the isoelectric point, 5, maybe 5.5, then we reduce the repulsion and we enhance absorption. Also, if we reduce disulfide linkages, so if we add a reducing agent and reduce 50% of the disulfide linkages, then that will enhance the flexibility of the protein and will enhance its unfolding and rearrangement at the interface. But additional thing here that is, this is in the native state, however, but you want to remember once you denature the protein, partially denature the protein without much polymerization, you really enhance its emulsification properties.
Sam,
Okay.
I have a quick question.
Yes, yes.
So you mentioned reducing disulfide linkages with like a reducing agent to enhance the emulsification. Is there like a food application or ingredient that could be used for this?
Yeah, like cysteine. Cysteine.
Oh, okay, okay.
Yeah.
Got it.
Cysteine can be used. It's food grade and it's reducing. It initiates redox reaction. Yeah. And also sulfites. I mean, sulfites can also be used. It's also food grade. Where was I? Gelation. Okay. So we talked about the gelation. It's mostly enzyme coagulation by breaking the glycomacropeptide by chymosis. You can induce heat coagulation, but you want to have a pH that is low and close to the isoelectric point. And if beta lactoglobulin is present and is denatured, it would complex with kappa casein and enhance protein-protein interactions. So the heat can induce that to happen under specific conditions. So, but mostly, as we said, the relation of casein is either enzymatic, acid, or age gelation, and we talked about that. Okay. For beta lactoglobulin, so it is thermally induced gelation, but it's also impacted by the pH. So if you have pH close to the isoelectric point, you form opaque. So because remember, the protein becomes non-clear, the protein solution will not be clear, so that you form opaque gels under these conditions. But if the pH is like, say, 6 or 7, mostly when we form gel, if it's just pure beta lactoglobulin, then you would form mostly transparent elastic gels. If you have high alkaline pH, net negative charge is way high, then it won't really form a gel for you. Or if it forms a gel, if you have high protein concentration and it forms a gel, the gel will be really weak. If you reduce the pH to around the isoelectric point, that's between 4 and 6, mostly we can say, let's say around 5 on average, you can start seeing gelation occurring even before denaturation. Why? Because again, you are really removing the net charge, and a little bit of heat above room temperature can cause thermodynamics, so that the protein can come in contact with each other at a particular protein concentration. So that doesn't mean that you can form a gel at 1% or 2% at pH at temperature 15 to 30 degrees. So protein concentration is important, and we'll talk about that in the next slide. If the pH is away from the isoelectric point and not really high, let's say pH 7, and that's where we mostly test gelation strength, around pH 7. So we denatured the protein first, so we heat it above the denaturation temperature so that the protein denatures, and then we can induce the formation of the gel. Heating rate is very important. So moderate to high heating rate, like 10 degrees C per minute, or increase in temperature per minute. This occurs at pHs that either under 4.5 or above 6.5. But if you have a low heat rate, like 1 degrees per minute, you can actually, at this very low heating rate, you can actually form a gel around the isoelectric point. But if the rates are higher than 10 degrees per minute, very high rate, instead of forming a gel, you end up denaturing the protein. A lot of thermodynamics, they come together, polymerize without holding a water, so without forming a balanced 3D network, so that means they aggregate, they precipitate. So if the system is formation of a gel at a particular pH that's around the isoelectric point, to prevent precipitation, we just have slow heating rate. So because the tendency to polymerize around the isoelectric point is higher, so if we want to form in a food matrix a gel around this type of pH, then we must be following slow heating rate. If it's high, then instead of forming a 3D network, you're going to cause precipitation. So other factors that impact gel formation is the salt concentration. And so if you have a little bit of salt, well, it depends on the protein, but the concentration varies, which concentration causes enhanced protein-protein interaction and reduction of repulsion. So a little bit of salt can help in formation of the gel. But high concentration can shield the charges like we talked about. It's kind of similar like salting out and precipitation. So we need to be able to find this sweet spot of the ionic strength for formation of the gel or enhance the gel formation. Sometimes you want to reduce the protein concentration that you're using for a gel formation, but in order to enhance protein-protein interaction, you have salt, so you can form a gel at a lower protein concentration. Then you can impact gel formation. If you want to prevent gel formation, you can block sulfidyl groups, or, which is not really commonly done, or you want to enhance disulfide interchange by adding cysteine, which is a reducing agent. So it basically reduces the disulfide linkage and frees sulfidyl groups that would trigger disulfide interchange. Protein concentration is very important, and we deal with that a lot in the lab when we want to determine what's the lowest concentration we can use to form a gel. And this is impacted by many factors. Sometimes you have a standard method where you go, okay, I'm going to use pH 7, no salt. What's the minimum concentration that I can form a gel? But sometimes you can manipulate and you can reduce the amount of protein that you need to form a gel by changing the pH and making it closer to the isoelectric point. But also, like I said, ionic strength can also help reducing the amount of protein needed to form the gel, not just the pH. You can add that to your notes. So at low or high pH, so yeah, once you have more charges, then you need higher protein concentration. Usually beta-electroglobulin or whey protein in general, you form a nice gel around 10%. A 15% protein will form a really strong gel. Okay, so moving on to production of protein ingredients, milk protein ingredients. There's so many pathways and there are so many ingredients out there. So of course we always start with milk, remove the fat, and or you don't have to remove the fat all the time. You can produce cheese from full fat milk, obviously, but formation of if we're looking at not cheese, just formation of protein ingredients, you skim the milk, you add rennet, you produce rennet, casein, and sweet whey, we call it. And or you can take the skim milk, reduce the pH, and then you form, you separate the whey by coagulating the casein, then you have acid whey in this case. And then you take the acidified casein that at the isoelectric point, you precipitate that, you can neutralize, and usually you neutralize with either, in industry, with either calcium hydroxide or sodium hydroxide. So you form calcium caseinates or sodium caseinates. So these are two types of ingredients that you would form based on what you use to neutralize the acidic casein or the precipitated casein. So if you heat the milk, the skim milk, you have calcium, you adjust the pH, you lower the pH slightly, and then you end up with a co-precipitate. So it's like all the proteins, casein and whey, are precipitated together. The reason we have calcium here is to help, and we reduce the pH a little bit to help with casein precipitating as well. The heat will cause the whey protein denaturation and enhances protein-protein interaction, even with the caseins. So you end up with a co-precipitate. It doesn't have much application, the co-precipitate, other than increasing the protein content and nutritional value of some applications. But it's hardly an ingredient that is used for functionality. So if we look at the whey, we have multiple ways that you can process the whey to produce protein ingredients. If you just take the whey and heat it to above the denaturation temperature of both alpha-lactalbumin and beta-lactoglobulin, you end up with a coagulated precipitate protein that is called lactalbumin. Again, it's not very functional, used mostly for enhancing nutritional properties by adding more protein. The main whey protein ingredients come from either membrane filtration. If you use only ultrafiltration, diafiltration, you end up with whey protein concentrate up to 80% protein. If you combine ultrafiltration, diafiltration with microfiltration, you get whey protein isolate. And this is, I'll show you the diagram to remind you. I've shown it before, but I'll show it again. And then the ion exchange is when you use ion exchange chromatography to produce whey protein isolate. And this is done in industry. Agripores, this is how they produce their whey protein isolate. And it's usually high purity, lower calcium, lower alpha-lactalbumin proportion compared to beta-lactoglobulin. So this is the diagram I showed before for the production of protein concentrate and isolate using membrane filtration. So remember here, you have the whey that comes as a byproduct from cheese production. If we do ultrafiltration, the protein will be in the retentate, and what goes through the permeate is your lactose and calcium, small molecular weight solubles, then you end up with whey permeate. The retentate then is your whey protein concentrate. Depending on how much you do diafiltration, you can change the concentration, increase the concentration of the protein and increase the permeation. So you will end up having more concentrate. That's why you will find products that are 34% or 35% protein, 50% protein, 80% protein. It depends on how much you wash and do diafiltration to concentrate your protein and remove lactose and calcium and other small components. So that's why it's more expensive to produce WPC, because it involves more water and processing and membrane processing. If we want to produce an isolate, so we take the retentate that we get from the ultrafiltration and we run microfiltration. In this case, your permeate is your protein, and what remains in the retentate is your phospholipids, large aggregates, protein that aggregated during the processing. So you would just end up with a very functional whey protein isolate that has about 90% or higher protein. So very briefly here, milk protein uses. So if we look at the lactic acid casein, so just the casein that we just separated by lowering the pH. Obviously, it's not soluble at pH 4.7, so 4.6, 4.7. It has low calcium and phosphate because at low pH, you have leaching of the calcium and phosphate into the whey stream. And you can solubilize it by increasing the pH. The rennet casein, obviously, it's insoluble at pH 7 because we remove the glycomacropeptide, so you don't have that stability. It has high calcium and phosphate. They're not leaching. You're enhancing the protein-protein interactions within the casein. In order to solubilize it, you can sequester calcium. So you can reduce the pH potentially, and calcium will leach out. And both are heat stable. Caseins are heat stable. Very good flavor. Nutritive value is high. Casein, well, that's protein efficiency ratio. But if we look at PDCAS, it's PDCAS of 1. It's used in different applications for texture, for flavor. So we can use it in breakfast cereals, snacks, baked goods. A lot of the use of casein is in the form of calcium caseinate, sodium casein. There's another form that I don't talk about is micellar caseins, but I don't have a slide on it. But anyway, calcium caseinates and sodium caseinates are common ingredients. Calcium caseinate is not very soluble. It forms a dispersion, but it's not very soluble. It's cross-linked. The caseins are cross-linked by calcium. They're dispersible, but they are opaque in color, and they have low viscosity because there's not a lot of interaction with water. So they're good for, because of their color and flavor and their dispersibility, they're good for whipped topping, creamers, imitation cheese. So these very specific applications. But the more functional ingredient is sodium caseinate. So it is very soluble and translucent in solution. It's a very good use for emulsification because it's soluble. So you want the protein to be soluble for emulsion as well. And it forms viscous solution. So it really is added to gravies, for example. I don't have it here on the application, but it added to gravies to enhance viscosity. It added to baked products like cupcakes, for example, or muffins. Added to infant formulas, nutritional beverages, meat products. So lots of applications. In terms of whey proteins, so you have the lactalbumin. It's not really functional. It's added for nutritive enhancement. And then the ones that are really functional are the concentrates and isolates. They're soluble. They are very common application is beverages, and they have good texture properties used in many, many applications, yogurts, processed meat, baked goods, snacks. And it says here they induce browning, in this case, especially whey protein concentrates because they have a little bit more lactose in them. So if in baked products, WPC aids towards the desirable browning through the Maillard reaction because of the presence of lactose. That's it for milk proteins. Okay, we have 5 minutes, and I don't think it's worth starting soy protein lecture unless you're very eager to start, not soy, I mean, legumes and oilseed proteins. Who would love to start this new topic now, and who would love to leave?
Just a question. Looks like after the calcium caseinate, sodium caseinate slide, looks like several slides were skipped.
Oh yeah, these were, don't worry about them. I had case studies, but for the sake of time, I was not gonna present them. Okay. I just want to make sure I cover the other types of proteins. That was back in the time when I, had more time, but the more I add material to my lectures, the less I have time to cover everything. So I chose not to cover these case studies. Okay, any other questions? No? I bet you would rather leave. Nobody said anything. Okay, I will start the legume and oilseed lecture next Wednesday. Okay. If no questions, see you guys next week. Bye-bye.
Dr. Bang, do you have a time that I could talk to you right now?
I had Ali requesting time, but how do you want to do that? Ali, do you want to go 5 minutes and come back?
Yeah,
I will leave the Zoom room open 5 minutes. I'll talk to Muaf and then you can come back.
Yeah, I can do that.
I'm sorry, Ali, I didn't know.
Is it related? If it's yeah, I can leave.
It's not related to the presentation. No.
Okay, I will leave then and I'll come back.
5 minutes, just 5
Okay,
minutes.
sounds good.
Okay.
I'm sorry for jumping in the last minute.
No,
I
that's
was
okay.
going to respond to your email yesterday.
Oh,
but,
hold on. I want to stop recording.